All right, so on to 13.4, Mendel's law of independent assortment. So in the previous section, we talked about the law of segregation, where the two alleles that a parent has for a particular trait gets separated during meiosis. And we were talking about a monohybrid trait, so a single or monohybrid crosses. We're talking about a single trait. We're going to get a little bit into dihybrid, di meaning two, and we can actually talk about the relationship for two alleles at two different locations on a chromosome. They can be inherited separately. This is really talking about even two different chromosomes. Um, so a dihybrid cross, we click on this here. If you haven't been doing that, it's excellent resource. So it's talking about um, two individuals who have alleles for two genes. It says heterozygous, but it doesn't really matter what they are. So a dihybrid cross, again, is talking about two different traits, two different positions on a chromosome. Really what happens is the alleles, they get sorted separately. So you have traits, especially particularly when they are on different chromosomes, they are not going to get inherited together. Um, and it talked about like flower color. So Mendel really talked about like flower color being inherited independently of plant height. So you weren't always having like purple flowers were not always tall. They could be tall or short. White flowers could be tall or short. So those are two different traits: the height of the plant, it being tall or short, and the color of the flower, purple or white, are two different genetic traits that can be inherited separately. And the law of independent assortment, alleles on one locus tend to be sorted into gametes independently of alleles in another locus or at other loci um, for that particular trait or those traits. Um, his results were published in 1866, and you have to understand that, again, this is groundbreaking stuff. I even kind of wonder if he really knew the impact he really, really had. I know the book says, he died in 1884. And he didn't know that his plants would be the starting point for modern genetics. He's often called the father of genetics because of this work that he did with the pea plants. But I often wondered, like, to what capacity he realized his impact would be. I mean, he was just making observations about various traits on pea plants, and to understand that certain traits would be inherited separately, that we now know is because they're on different points of chromosomes. To really have seen that and to kind of explain that in his time is a huge deal. Because again, at his time we didn't know about genetics in the way we know now. We didn't know about DNA being the way that genetics is stored and transmitted from one generation to the next. But it's a huge deal to be able to look at this to see. And I like to kind of look at this, and this is talking about independent assortment in the terms of looking at meiosis. So again, just like the law of segregation, independent assortment refers to what's going on in meiosis. And I actually mentioned this when we discussed meiosis about how how the chromosomes, the chromatids, get independently put in different chromosomes. They get randomly put. So I'll make this a little bit bigger, hopefully. I don't block too much here. Ooh, I did not mean to do that. So, let me make me a little bit smaller there. So we have this particular this particular cell right here, and you can see the chromosomes. This is just, showing a couple pairs of homologous chromosomes. We have pink and blue. They've been duplicated, which is why we have the X, the pink with a little bit of blue, the pink with a little bit of yellow, the blue with the darker blue, and then the blue with the orange. And we have 2 copies of each. So then we have what happens in meiosis 1. They're getting linked up. They will get split. So even from this point, we got letters attached to them. So let me click back over to here. So you see this one has a P, and this one has a T. So we're looking at 2 different traits. And during meiosis 1, when those get split, you have this side going over here, this side going over here. And it could have happened the other way. You could have had, the blue chromosome with one of the pink. This one could have gone with this one. But it happens randomly. It happens independently. And then you see in here at the very end, here, you have a big, see, this is why I don't like P's. You have a little p and a little t, a big P and a little t, a big P and a big T, a big P and a big T. And this is just how it happened to separate in this particular example. However, we can look at the opposite. We can look at, it can happen this way or it could happen this way. I don't know if you notice this little or right there. So it could happen this way, but maybe the next time gametes are made, they get separated a little differently. So because the trait for P and the trait for T are not linked, they're not on the same chromosome, so they can get separated differently each time. So in this case, we have the pink with the P and the blue with the T over here, and the blue with the P and the pink with the T over here. So these 2 different traits are going to be inherited differently. And we can see that there's a bunch of possibilities. And this is one of the ways that meiosis contributes so much to genetic diversity, is this independent assortment, that these genes on different chromosomes get randomly put into the gametes. And it's not always going to happen this way. It's not always going to happen this way. There's going to be some changes in that. So when we, talk about meiosis increasing genetic diversity, like I had said before, these get put in there randomly. It just depends on how they're sorted. And then again, which ones get fertilized, because even though these are possible gametes, it doesn't mean that those are all going to get fertilized. And as the case of the ova being made, remember there's 1 egg cell with 3 polar bodies. So if we were looking at in the female flower and an egg cell was being made, only one of these would be possibly fertilized each time to produce the next offspring. In plants, the polar bodies get fertilized too, but they don't become new members of that species. They wouldn't result in new plants. They help out with feeding the developing embryo. However, it kind of goes about, then in your book here, it talks about genes on the same chromosome. So this is talking about genes on separate chromosomes. And when Mendel was actually studying, again, he didn't know anything about chromosomes. He just knew what he could see. Those 7 genes that he was looking at, they sorted independently. And it just so happens we now know that the genes he was studying were actually on the same chromosome, but they are far enough apart on the chromosome that they can get exchanged during crossover. If you remember, crossing over happens during prophase 1 of meiosis 1, and it's when these homologous chromosomes match up and they swap genetic information. So the further apart they are in the chromosome, the more likely they are to to cross over and be swapped. But if they're closer, they're usually not switched out. I wish your book. Let me see if I can make a quick little. So if we have a chromosome, like if this is your chromosome, let's just say, and you have, these are your different gene locations here. Then your likelihood of them being separated depends on how far apart they are. So if this is our gene, we're just going to call this gene A, gene B, gene C, and gene D. Or really, I should say locus A, locus B, locus C, locus D. The closer they are, the less likely they are to separate. So A and B are very close. They are less likely to be separated. It doesn't mean they are never going to be separated. They are less likely. Whereas A and D are much further apart. They are more likely to get separated during crossover. Same thing with, C is more likely to be associated with A than D is because C is closer to A. So the closer they are, the more likely they are to stick together. But again, it doesn't mean it's impossible because during crossover, this gene could be swapped out and these could be remained. And again, that's another thing that increases the genetic diversity in sexually reproducing organisms. All right, I think that is a good place to stop for this section. That was probably a lot of information.